Okay, hello, I'm Dr. Curtis. I am sorry I couldn't make the meeting to describe the various projects that I have in the lab, but I did want to actually give you a video so you can get some idea of the things that I was talking about in case there's someone within the group who's interested in applied biotechnology type project. Over the past 25 years, we've been working in various areas in plant biotechnology, working in plant propagation as well as production of therapeutic proteins. Metabolic engineering has included the actual expression of membrane proteins, as well as the placement of biosynthetic pathways for hydrocarbon production into alternative hosts. And I'll be describing a specific project that I'm offering for this applied master's type program in the area of production of biopolymers in cyanobacteria. A lot of our work focuses on the basic area of bioreaction engineering, including aspects of both the design as well as process control and operation. And I'll also then be describing work around the idea of producing co-products associated with biofuels. The time in this lab is somewhat unusual. Over the past a year, we've had 5 PhDs that have graduated. As a result, there's projects that have some loose ends. At the same time, a lot of the methodologies are good to go as far as learning those. And so it is a good time for this type of an applied project. Mustafa actually did work in aquaporin type work and membrane expression. Jen was working in algae biofuels. Naimal was working on genetic engineering of hydrocarbons, and cellulosic biofuels is what Trevor was doing. And Sergio was working on applied plant propagation. The 2 projects that I had put together that I thought would be particularly appropriate for this program are both related to protein expression. The one is expression of an embryogenic growth factor that will induce embryogenesis in plants. The other one is production of a bioplastic polymer in there. In addition to that, a large fraction of our work focuses around biofuels and bioenergy production. And so if there is interest specifically in that area, I could certainly put together something that has a little more of that focus. The first project then is looking at protein expression for embryogenesis. It's related to plant biotechnology and in particular protein engineering. The specific work would involve working in the pilot plant. This is a facility in the basement of Penn. For over 10 years, I actually helped run this facility, including the operation of large-scale pilot fermenter systems. And so that particular project would involve learning how to run these for the production of a specific protein. This skill set would be directly applicable to biofuels fermentation industry as well as pharmaceuticals production. And it represents a pretty unique pilot plant experience in a $20 million facility for that technology downstairs. The scientific basis for this project is work that we've done. First was with Weyerhaeuser and tree propagation, where you can actually clonally propagate trees and take a superior tree and clone it. Whereas if you take seeds, you actually end up with high variability. And so this is actually a commercialized process where they actually can take that and make the embryos in suspension culture, and then from that, plate that out, and they actually have a process developing artificial seeds for production right now. We've expanded this to many other companies that we've worked with, including DuPont, Pioneer, and others. Working with various reactor systems. This is a wave system used a lot in the pharmaceutical industry right now for CHO work, Chinese hamster ovary, and antibody production. And a lot of other unique reactors. This is a roller carboy. This is a temporary immersion reactor where you actually lift the reservoir and flow the media into the reactors. And a characteristic you'll see is we do quite a bit of instrumentation and unique reactor design type work. The basic science behind this, the most recent work by Sergio, was to deliver transcription factors. These transcription factors actually then kickstart the embryogenic process. So then by taking this and using this vector, agrobacterium, and inserting that into a leaf, it literally makes the leaf cells think that they're embryos. And from that, you can actually then do massive propagation of clonally propagated trees. This work did involve quite a bit of molecular biology, including the cloning of the genes, and successful work with bioinformatics to ultimately take that and genetically engineer it into the material. And that genetically engineered material, you can see here, dramatically improved both the quality and the quantity of the embryos that were being formed. This was a project actually for disease-resistant cacao, so it was a project to assist in the disease problems they're having right now with chocolate production. There's another protein that is specifically for this project that I'm proposing for this. It's called the PLA proteins, or cyanin-like functional proteins. Why we're so excited about this is it turns out that it's an internal domain that's functionally active. And because that internal domain does not have any sugar groups on it, it can be expressed in bacterial systems, which allow it to be produced cheaply. And so we have about a dozen biotech companies that are interested in having this material to test it in their specific species like rice, wheat, corn, etc. And so that would, the objective of this project would literally be to produce that material and then distribute it to the companies for their interest. Preliminary results here showing a cotton version of this, and that cotton version actually applied to tissue that was from the chocolate tree. You can see a very enhanced and improved appearance of the embryos that were forming. And so we're interested in now producing enough of this material by larger scale techniques, not just simply His tags, but also tangential flow filtration to purify that material. If you want more background on that, you can just type baby boom into that. That is the transcription factor in Curtis, and you'll find this paper on the use of this that was just published this year. The second project then is to do with algae-type biofuels, and in particular the production of a bioplastic protein as a co-product. And this particular bioplastic, it turns out, okay, first is what work involved. It's a little bit of molecular biology, and hopefully we're planning certainly by summer to have this so that we can take it down to Mesa, Arizona to this algae testbed facility. And then that facility will allow us to do protein expression in large scale systems. These are actually 3 of my students down in, a couple of months ago down in Arizona, where Jun is actually on site after his PhD doing something related to a startup. The skill set then has, from molecular biology moving to spud reactor scale up and elements of process control that I'll describe very quickly here in a minute. The protein of interest, it turns out, comes from a squid. So it turns out within the squid sucker, there's actually a fingernail. That fingernail is actually made out of material that's very much like spider silk. And so that ring tooth makes a bioplastic, which is thermoplastic, so that you can actually do extrusion and other aspects of that. And that gives us a basis then for producing it by taking the genes that make this particular protein, squid root SRT, squid ring tooth, and expressing it in other systems. That was actually done by various techniques of molecular biology. We ultimately then defined that. In fact, we now have half a dozen genes that make these from different squids or same squid. They make multiple different forms of this protein. We've expressed it in the pilot scale systems that I mentioned in the previous project downstairs in genetically engineered E. coli. The problem with E. coli is it uses sugar for fermentation, which is costly, so we're much more interested in a cheaper feedstock, which would be use of cyanobacteria and carbon dioxide as a means to turning that to polymers. How this works in cyanobacteria is you actually have a basis for homologous recombination rather than the typical genetic engineering that you may have learned from scissoring, if you will, cutting and pasting DNA. Homologous recombination means you take advantage of the native plasmids that are in there, not the chromosome. And then you have a region here that's the same as you'll find on the native plasmid, and by introducing this DNA, that gene of interest, in this case the SRT, will actually move into there, and then you'll have multiple copies of that gene for expression with that protein in the cyanobacteria. It turns out this technology was developed by Don Bryant at Penn State, and it's considered to be the highest protein expression system yet developed for algae or cyanobacteria.
Stop for a second.